Enzyme Kinetics, the second lecture of the series. Factors influencing the enzyme activity are temperature, pH, substrate concentration, and enzyme concentration. Temperature dependence of enzyme activity and pH dependence also we had discussed earlier in the last class with an emphasis as to why we get shapes of the curves like this. For example, temperature, as you increase the temperature, the enzyme activity increases almost linearly till optimum temperature where it reaches the peak. And thereafter, typically after about 40 to 42 degrees Celsius, there is abrupt fall in the enzyme activity. This is because of denaturation. So the first phase, the rise was because of increased kinetic energy of the substrate molecule, and this second precipitate fall is because of denaturation. Likewise, we had seen when we plotted pH against the reaction velocity, what we obtained was a bell-shaped curve. Optimum activity here, change in the pH on either side results in drastic fall of the enzyme activity. And as we had discussed, this is because of change in the charge status of the amino acid residue at the active site, because charge on an amino acid depends on pH of the surrounding medium. Now, one thing, the optimum pH for various enzymes that varies. Optimum temperature, as we said, for all the human enzymes in our body is nearly the same, the body temperature, but pH, optimum pH varies. It may be in the acidic range or in the alkaline range, but most of the enzymes, they function optimally at the physiological pH. Now we come to the third factor, that is the substrate concentration. How does it influence? And as we will see in this curve, that what we obtain is a rectangular hyperbola. You can see in the blue line, a rectangular hyperbola. You in incubate a fixed amount of enzyme with various varying substrate concentration and observe change in the reaction velocity. And as we can see here, initially there is near linear rise in the reaction rate, the velocity, by increasing the substrate concentration, but it becomes more and more flat till it reaches a peak, and no further rise in the reaction velocity is reached by increasing the substrate concentration. Why? Why of this is most important, what we are going to discuss. Now, what happens is that initially, as you add the substrate, the it immediately finds an enzyme active site to bind with, because the enzymes are present in uh, adequate amount now. But as you add more and more substrate, more and more active sites become occupied. And most of the enzymes now are present in form of ES. They are present in form of bound with the enzyme. So most of the active sites are now occupied, so the reaction rate slows down. And when all of them are occupied, then a peak is reached, which is called Vmax, maximum velocity. So now, if you increase the substrate concentration, the reaction velocity will not increase. I can compare the whole scenario by ticketing counters at a busy interstate bus terminal. During the morning hours when the passengers are scarce. So that time the number of passengers increase hoga, ikka dukka aa raha hai, immediately he will find some booking clerk, some empty booking window, and he will get the ticket. So number of tickets sold per minute will depend will be directly proportional to number of passengers. But during peak hours, when the number of flood of passengers is coming and a long queue has been formed. So that time, it is not the substrate concentration. I mean, it is not the number of tickets sold, let me say, is not proportional to number of the passengers. It is, you can say, on the efficiency of the booking clerk, but that also has got its own limitation. So that time, irrespective of how long is the queue during rush hours, number of passenger, uh, I mean, number of passengers does not determine number of tickets sold. I think I have taken too long on this. So let me return to it because at this point, saturation point, this we called actually saturation kinetics, because active sites are more or less occupied, all enzymes are present in the substrate bound form. In the above example, if you what if you increase the number of the ticketing window, if you increase the number of the booking clerk, if you double the number of the booking clerk, don't you think that number of tickets sold will also be doubled? Likewise, if you increase the enzyme concentration, more active sites will be available and more substrates will be able to bind with them. So therefore, the reaction velocity will increase proportionately with the linearly with the enzyme concentration. So enzyme concentration, if you increase a linear relationship, substrate, then a rectangular hyperbola. I repeat, rectangular hyperbola, initially it is first order kinetic. Later, when Vmax is reached, that time it is zero order kinetics. Just to introduce one term, Km. This is half of the maximum velocity, and this reflects affinity of the substrate for the enzyme. But this we will talk in some other video. In this video, I have tried to explain in this one and the previous one why different shapes are obtained. Bell shape, rectangular, hyperbola, linear. Because, I found that the students are able to draw the curves, name the curves, but why we obtain these curves? There they fumble. Hence this video. Thank you so much.